Okay, good morning to everyone, and thank you for the organization to the organization for inviting us to present our work here today. Today I'm going to present a work we did, which is showing that Lewy body fractions from patients with Parkinson's disease can initiate alpha-synuclein-dependent neurodegeneration in mice and non-human primates. As you all know, Parkinson's disease is mainly characterized by the dopaminergic cell loss in the substantia nigra pars compacta and the presence in affected brain regions of insoluble cytoplasmic inclusions, mainly consisting of alpha-synuclein, which are the Lewy bodies. This alpha-synuclein in Parkinson's disease and other synucleinopathies is mainly deposited in a hyperphosphorylated form with a fibrillar structure. Multiplications and also 3-point missense mutations have been so far identified in familial forms of Parkinson's disease and also in disease with Lewy bodies, pointing out that abnormalities in alpha-synuclein can be causative of these diseases. But despite all this big research that has been done in this sense, we still don't know what is the exact relevance of Lewy bodies into the pathogenic process of Parkinson's disease. In the last years, there is mounting evidence that neuropathological alpha-synuclein lesions in Parkinson's disease can self-propagate and might spread progressively through interconnected brain regions by a cell-to-cell transmission mechanism. Supporting this concept, Bragg proposes a model in which Lewy body pathology can spread from rostrocaudal axis from the neuronal nuclei in the medulla oblongata in early stages of the disease to mesencephalon and neocortical areas as the disease progresses. It's true that this model remains controversial because it cannot explain all the pathological cases that have been found. However, what we want to point out is that alpha-synuclein pathology does not appear at the same time in the whole brain. On the contrary, it seemed to progress somehow between interconnected brain, anatomically interconnected brain regions. In this line, Cordova published a beautiful work in 2008 using embryonic neurons that were transplanted into the striatum of an individual with Parkinson's disease. These grafts survived for 14 years, and they were viable, expressed in tyrosine hydroxylase, BMAT, but not dopamine transporter. The interesting point in this data was that not only in non-grafted host original Parkinsonian brain, but also in the new engrafted neurons, there was alpha-synuclein, there were alpha-synuclein inclusions that were positive for ubiquitin. So this was suggesting a host-to-graft transmission of the Lewy body pathology in the human brain. The SPLAT further explored this mechanism of progression or transmission of Lewy body pathology in the brain, and they show that alpha-synuclein can be transmitted from neuron to neuron via endocytosis, and this alpha-synuclein can form Lewy body inclusions in the cells. They did intrahippocampal injections from mouse cortical neuronal stem cells in an alpha-synuclein transgenic model. The stem cells were infected with GFP, so they were labeled in green, and the alpha-synuclein were labeled with tyramide red. So they could show how this alpha-synuclein could be uptaken by the engrafted neurons and internalized, as you can see here in the merged images or here. Furthermore, this alpha-synuclein was able to form inclusion bodies in the cells that could be quantified and show almost a 50%, they were almost present in the 15% of the cells. And they also studied the intracellular distribution of this alpha-synuclein using a cell model which was consisting of differentiated SHSY5Y cells that were overexpressing alpha-synuclein and were co-cultured with the same cells not overexpressing alpha-synuclein. And they could see how the alpha-synuclein was transmitted from the donor cells to the acceptor cells in two different ways, either with a diffuse, kind of diffuse accumulation of alpha-synuclein, or also with the formation of inclusion bodies. These inclusion bodies were positive for ubiquitin and also for the amyloid binding dye thioflavin S. And what was more interesting was that the transference in both cases, when the alpha-synuclein was present diffusely or when it was forming inclusion bodies, the amount of alpha-synuclein internalized by the acceptor cells was depending on the alpha-synuclein expression level in the donor cells. A little bit more recently, the laboratory of Virginia Lee showed that a single inoculation of synthetic recombinant alpha-synuclein fibrils into striatum of mice brain, either wild type of alpha-synuclein, was able to initiate an alpha-synuclein pathology that could be transmitted to interconnected brain regions. As it's shown here at 30 days, well, in the striatum, which was the injection site, and also in the cortex. And with the time after the injection, this pathology was even stronger and could even be seen in the contralateral cortex. Inside these cells, there were also inclusion bodies that were positive for hyperphosphorylated alpha-synuclein. This pathology was also accompanied by dopaminergic neuronal loss in the substantia nigra, which was progressive, and also accompanied by motor deficits and reduced levels of dopamine in the nigrostriatal pathway. And even if they were taking brain extracts from old mice that were presenting alpha-synuclein pathology, and they were injected into young asymptomatic mice, these brain extracts were accelerating the initiation and progression of the Lewy body pathology. Oh, well, I would like to also say first that Matsuda in the last years also published and showed that these synthetic recombinant alpha-synuclein fibrils injected into mice brain could trigger the pathological conversion of endogenous alpha-synuclein from the host murine cells. But despite all the work that has been done, which is a lot, there are still so many questions that remain unanswered. And we were especially interested in knowing if human alpha-synuclein that is normally contained in the Lewy bodies and is linked to Parkinson's disease could have this potential to cause neurodegeneration in this type of models. And also, if it can cause the same pathogenic effect that it was seen for synthetic recombinant alpha-synuclein fibrils. And finally, if this process that has been shown in rodents can also occur in species closer to human. So for this, we designed a number of experiments to explore the pathogenic effects of these negral Lewy body extracts from PD patients by inoculating them into the brains of wild-type mice or macaque monkeys. For that, human substantia nigra was dissected from fresh frozen post-mortem midbrain samples and from 3 different sporadic Parkinson's disease patients that were presenting Lewy body pathology. The pathogenic alpha-synuclein was purified by a sucrose gradient, obtaining 20 fractions from top to bottom that were analyzed for the presence of aggregated alpha-synuclein. The analysis that we did was a filter retardation assay that allows to detect aggregated alpha-synuclein by the ability of a membrane to retain detergent or urea-insoluble amyloid fibers or high molecular weight aggregates. And as you can see here, these would be the 20 fractions that we obtained from the sucrose gradient. This would be fraction 1 to 10, fraction 11 to 20, and most of the aggregated protein was contained within the fractions 15 to 19. So these fractions were the ones that we used to mix them all together in the same proportion for injection in animals. As a control, we used two things. One thing was the vehicle, which was the corresponding sucrose fraction, but without any Lewy body sample inside. And the other one was what we called a non-Lewy body fraction. We used the fraction 6, but we could have used whatever from 1 to 9, more or less, which was coming from the same patients, but didn't show any aggregated alpha-synuclein. These samples were also quantified for human alpha-synuclein using a specific ELISA, and we saw that the amount of alpha-synuclein that they contained was approximately 10 picograms of alpha-synuclein per microliter of sample. When we analyzed these samples by electron microscopy, we saw that there were presenting amorphous or spherical aggregates with a filamentous structure that was positive for alpha-synuclein, as it's shown here in the immunogold. But we thought it was not so suitable for our injection in animals because the aggregates were so big. So what we did was to bat sonicate these samples for 5 minutes prior to injection, resulting in the disruption of the aggregates into fibril fragments of different sizes. To further corroborate that the aggregated alpha-synuclein that we were observing in this filter retardation assay, we did immunoblotting for hyperphosphorylated alpha-synuclein, ubiquitin, and P62, all of them known components of the Lewy bodies. Furthermore, alpha-synuclein could be shown by fluorescence microscopy to co-localize with the amyloid binding dye thioflavin S. So we used adult wild-type mice to inject either the Lewy bodies extracts, the non-Lewy body extracts, or the vehicle in one single unilateral injection just above the substantia nigra. And these brains were analyzed at different time points for 24 hours to 17 months to study the nigrostriatal pathway and check the integrity. This was done at two different levels. At one level, we studied the integrity of the dopaminergic neurons in the substantia nigra pars compacta by a stereological cell counting of the tyrosine hydroxylase positive cells. And in the other side, we also analyzed the integrity of the striatum by optical density of the striatal Th positive fibers. There was no evidence of nigrostriatal degeneration in the first time point for weeks, but after 4 months post-injection, we could already see that there was an ongoing progressive neurodegeneration in the fibers from the striatum, which was going up to 17 months. Concomitantly to this striatal neurodegeneration, there was also neuronal cell loss in the substantia nigra, but only in some mice, that was more extensive and applying to all the mice studied at 17 months. On the other hand, no signs of nigral neurodegeneration were observed in the mice injected either with the vehicle or with the non-Lewy body fractions. These cells also show signs of astrogliosis, as it's shown by the staining of EVA1 positive cells and GFAP astrocytic reaction. And they also had motor deficits. Here is the Lewy body injected mice compared to the vehicle injected mice or the non-injected mice that we did by a poll test. So to further assess if this nigrostriatal neurodegeneration that we were observing in the Lewy body injected mice was correlating with an alpha-synuclein pathology, we used an alpha-synuclein antibody that was detecting human alpha-synuclein but not murine. And we could see that the external alpha-synuclein coming from the Lewy body extracts was already internalized in the mice cells at 24 hours, also in Th dopaminergic neuronal cells. And the immunostaining appear mostly as a punctate inclusion-like staining. But this human alpha-synuclein that was detectable at 24 hours could not be detected at later time points. However, when using an antibody that recognizes both human and murine alpha-synuclein at 4 months, we saw a big signal in Lewy body injected mice, which was corresponding to dopaminergic Th-positive neurons that could be only attributed to the endogenous alpha-synuclein from mice, because we couldn't detect any more human alpha-synuclein. So this is telling us that the exogenous alpha-synuclein that we injected, that it's coming from the Lewy bodies, was triggering the pathological conversion of endogenous alpha-synuclein in the mice. To further confirm this hypothesis, we observed by immunohistochemistry for alpha-synuclein before and after proteinase K digestion, that whereas in the non-injected mice, no alpha-synuclein signal was remaining after digestion with protein kinase, in the Lewy body injected mice, there was some signal that was resistant to this digestion. And you can see here what's the difference between the Lewy body injected mice and the non-Lewy body injected mice, or the vehicle injected. So this is mainly telling us that this endogenous alpha-synuclein from the mice that have been converted into a pathological conformation, let's say, by the exogenous alpha-synuclein, was adopting an aggregated or beta sheet conformation. To further corroborate this pathogenic nature of alpha-synuclein, we did an staining for hyperphosphorylated alpha-synuclein, and we could observe a signal in the Lewy body injected mice. This signal could not be detected in the latest time point at 17 months, probably because cell death is already fully established at this point. And also what it was very interesting was that hyperphosphorylated alpha-synuclein could also be detected in other brain regions connected to the injection site, such as the striatum or neocortical areas, where the Lewy body injected mice showed much more hyperphosphorylated alpha-synuclein than the non-injected mice. But the Lewy bodies contain many other components besides the alpha-synuclein, and we wanted to be sure that the pathogenic effect we were observing was due to a specific process or activity by the alpha-synuclein. So we used these original Lewy body extracts, which are shown here, to do 2 sequential immunoprecipitations in order to deplete alpha-synuclein from these samples. We took the supernatant of the second immunoprecipitation, and this supernatant was injected into mice and compared to the Lewy body injected mice. What we saw was that whereas the Lewy body injected mice were showing an increased hyperphosphorylation of cells, the alpha-synuclein depleted mice were not showing an increased phosphorylated alpha-synuclein compared to the non-injected mice. Also, this pathogenic process seem to be dependent or seem to need the host expression of endogenous alpha-synuclein, in this case murine synuclein, because when the Lewy body extracts were injected into alpha-synuclein knockout mice, these mice, which are shown here in these gray bars, didn't show any sign of neural degeneration or alpha-synuclein pathology. However, we found these results interesting in murine. What we wanted to be sure is that this process was also occurring in some species more relevant to human disease. So we decided to use monkeys that were injected at 2 different points. 2 monkeys were injected into the striatum and 2 monkeys were injected into the substantia nigra. And they were analyzed at different time points by PET for the density of TH fibers in the striatum. We could see that already after 9 months, these monkeys were showing reduced levels of striatal innervation that was sustained till 12 months. This was confirmed by neuropathological analysis, where we could see that the monkey injected into the striatum had about a 30% reduction in the striatal innervation. This was also happening in the monkey injected into the substantia nigra. And concomitantly to this neurodegenerative process in the striatum, there was also a 40% reduction of the neurons in the substantia nigra in the monkey injected into the striatum, and a 15% reduction of neurons in the substantia nigra of the mouse injected into the substantia nigra directly. This is telling us that the disease progression in the mice injected directly into the substantia nigra is progressing slower or is not such so pathogenic as the one injected into the striatum. This neurodegeneration was also analyzed at the level of pathogenic alpha-synuclein by immunohistochemistry, and the mouse injected into the striatum was analyzed at different regions of the brain to see how the expression of alpha-synuclein could be spreading through these interconnected brain regions. We could see that the mouse injected into the striatum was showing an increase in alpha-synuclein expression, not only in the injected site, but also in the regions that received input from or that were sending afferent connections to the injection site. Such was the caudate and the putamen, the globus pallidus, the substantia nigra, the prefrontal gyrus, the entorhinal area of the temporal cortex. Besides of the monkeys that were injected into the striatum and the substantia nigra, we also wanted to know what was the contribution of the nigrostriatal pathway to this putative spreading of the disease. So one monkey from each group, so one from the striatum and one from the substantia nigra, were pre-treated with the neurotoxin MPTP to destroy the nigrostriatal pathway and to see what was the potential contribution of this pathway to the disease progression. So the monkey, which was injected or treated with MPTP prior to injection, showed only a local accumulation of alpha-synuclein in the injection site that was projecting to the efferent areas but was not accumulating retrogradely. So no expression of alpha-synuclein was observed in the substantia nigra. On the other side, the monkey which was injected into the substantia nigra was only showing local accumulation of alpha-synuclein in the injection site, here in the substantia nigra, but not so much spreading through the interconnected brain regions. Also, the MPTP mice was showing, anyways, accumulation in the substantia nigra. So overall, our data suggests that Lewy body linked human alpha-synuclein, which is derived from Parkinson's disease patients, can be pathogenic in both rodent and non-human primates. And this derived alpha-synuclein can induce the pathological conversion of endogenous alpha-synuclein from the mice, which is associated to a progressive neurostriatal degeneration, which is mainly starting in the striatal terminals and later on going into the neuronal cell bodies, which is exactly what is observed in the Parkinson's disease patients. The Parkinson's disease-like pathogenic effects are dependent on both exogenous alpha-synuclein, which is present in the Lewy bodies, but also depend on the host endogenous alpha-synuclein. And finally, Lewy body-induced alpha-synuclein pathology can disseminate transsynaptically because the majority or the vast majority of the alpha-synuclein signal that we could observe was in the synaptic terminals, and can disseminate both anterograde and retrogradely to anatomically interconnected brain regions which are distant to the injection site, with a more limited spreading when injected into the substantia nigra, which is probably due to the higher connectivity of the striatum. I only want to show just very quickly that this project is not finished here, and we want to, there is already an ongoing project in baboon monkeys using 53 monkeys, so it's going to be a very robust study, which is financed by the Michael J. Fox Foundation for the next 3 years, and in which we want to explore the kinetics of this prion-like Lewy bodies-induced neurodegeneration, and to check also the specific vulnerability of dopaminergic neurons to uptake and propagate this alpha-synuclein aggregates. We are going to monitor the symptoms through the whole experimental setting, and we also want to study the effect at the central nervous system level of the administration of these Lewy bodies that we are going to use also with these monkeys, either injected into the striatum or into the prefrontal cortex. We also want to administer these Lewy bodies in the ventral wall of the stomach and duodenum and see how is the spreading of Lewy body pathology, if there is some spreading at all, along the central nervous system. Just to finish, I want to mention all the people that has participated in this work, especially Ariadna Recasens, who is the main author of these results, and also Jordi Bové and Benjamin Dehaye from Bordeaux in France, the lab from Isabel Fariñas, Ana Pérez, the lab from José Obeso and Blesa, and I also wanted to mention the University of Murcia, the lab of Teresa Herrero, that didn't participate in these results that I showed today, but they are doing a great effort already organizing and preparing the colony of baboons that it's going to be used for the study in monkeys. Thank you very much.
Thank you. So we have time for questions. There is one there on the back.
Very nice talk. Let me ask you something. Probably I misunderstood you. You said that the concentration of synuclein within those preparations from Lewy body was 10 picograms per microliter?
Yeah, well, actually this is something that we have to discuss a little bit more. The thing is that we use a specific ELISA for human alpha-synuclein in these samples, and this was the concentration that we obtained from this ELISA, which is about 10 or 20 times lower than it was reported with the synthetic alpha-synuclein. But the impression we have is that this ELISA is only measuring soluble alpha-synuclein and not so much aggregated alpha-synuclein, because well, I don't know if I can see it now, but if you see the images I have from the filtered retardation assay, where I have the concentration of alpha-synuclein in Lewy body fractions and in non-Lewy body fractions were similar. So for that and for other results we have from Western blot, we suspect that the ELISA is only taking into account soluble or monomeric alpha-synuclein and not aggregated alpha-synuclein.
Well, anyway, you could make it soluble and then check it. Okay, the other two questions, short ones, is referred to the controls you make. Actually, the control you make for an injection, I would like to see better than that, is just to take your fraction that is the positive one, ultracentrifuge in an air fouche, and take the superrate, and that should contain no synuclein, and inject that to see whether it's still producing the same effect. That will be a good control for your experiments, better than treating with proteinase K or with any other things. And another question respect to one of the films I didn't see it quite well. How far was the proteinase K resistant synuclein after injection from the site of injection? Those small dots that you saw in the...
Sorry, I didn't hear the beginning of the...
You saw on a slide that there is some proteinase K resistant brain structures.
Yeah.
How far was that structure from the site of injection of synuclein? Was it close or was it far away?
I cannot remember exactly, because the injection was in the substantia nigra, and these proteinase K resistant alpha-synuclein deposits were also in the substantia nigra, but I cannot remember exactly how far they were from the injection site. I'm sorry.
Okay, thank you very much.
So, justo.
Very beautiful work. Thank you very much. My question is not related to your work. I mean, it's just indirectly related. It's a question of a clinician, so forgive me if it's not appropriate. The issue is, if there is propagation of synuclein pathology, why it does not follow anatomical connections? For instance, I mean, after the substantia nigra, we should see synuclein pathology in the striatum, but we do not see Lewy bodies in the striatum of patients with Parkinson's disease. Why is that?
Well,
what are the connections between the olfactory bulb and the locus coeruleus or the nucleus raphe magnocellularis? What are the connections between raphe magnocellularis and locus coeruleus with substantia nigra?
Yeah. Well, if I could answer that question, I would be publishing it, because it's the big controversy right now in the field. It's also the same controversy that we have with the BRAC model. I think it's difficult to explain, and this is something we all have to work in. But probably the fact that this pathology is progressive, is being transmitted not only retrogradely but also anterogradly. This is including an extra effect, because besides having, for example, alpha-synuclein pathology in the substantia nigra that it's going to the striatum and so on, so this alpha-synuclein pathology would be moving back and forward to different brain regions. And also, I guess we have to take into account that each patient may have, I don't know, genetic alterations, anatomical alterations that might be modifying specifically in each one how this disease is progressing in the brain. But the truth is I cannot answer that question right now.
I have a question myself. You are recapitulating the advance in the field of in the prion protein, and you are seeing cross-species conversion and also the transmission even from the GI, from the gastrointestinal tract. So that made you guys kind of worrying. Are you suspecting that it might be some synuclein deposits in the cattle, in the animals that we eat, and there might be a way of let's say, contaminant or the toxic hypothesis of Parkinson's disease might be an origin of the disease also there?
In principle, we don't take that into account. We're working with animals which are supposed to be equal ones to each others, and using all the controls we have in our hands. So in principle, we have to attribute the differences we are seeing to the differences in the injection that we are administering to these mice. But I don't know how a specific toxin could be acting on these.
But is there any evidence of Lewy bodies in other species, in animals?
You mean in other species? Well, I'm not sure, because I don't think there are so many Lewy bodies in other species, but I'm not really sure about this. Probably non-primate humans, but normally these kind of aggregates are mainly present in humans and not so much in other animals.
Okay. So if there are no more questions, yeah, there is one more there.
Congratulations for the very, very nice work. The MPTP models, basically done about 20 years ago, had a clear age-at treatment or exposure effect, which was attributed to the melanization of substantia nigra, which was a progressive.
Sorry, could you put the...
Yeah, I think the MPTP models had a clear age-at treatment effect, which was attributed to the melanic deposit, which was not from birth. And there are also epidemiological data suggesting that there are childhood infection linked to Parkinson. Did you observe any kind of age at treatment effect, or was this...
No. The problem with the MPTP treatment was that it was done only in a small number of animals. So we could not compare different ages. We were only administering MPTP at one point, and we couldn't distinguish age effects. However, now that we have this big study in baboons, this is something we could do, because obviously we are interested in using all the controls that we couldn't use in the previous study, and into going a little bit further in the study of the pathology.
Okay, so thank you. I think this is it.